So welcome once again to the course Next Generation Sequence Analysis here at NYU Poly. My name is Dr. Jonathan Flowers. I'll be your instructor for the course. Hope you got a chance to check out the introduction and welcome video, which is just a short clip where I introduce myself a little bit more. So today we're just going to dive right in to what I think is pretty important for doing good next generation sequence analysis and good bioinformatics, and that is to know a little bit about both the methodologies, the biology behind the bioinformatics, and to know a little bit about the machines you're working with that produce the data that we'll be analyzing. All of this goes into reducing errors, better understanding the data, and just doing a better science. So I'd like to start here just by telling you a little bit quickly what next generation sequence analysis is. So essentially about 5 years ago, some of the technology really shifted to going from a capillary-based sequencing strategy to what we call massively parallel or short read sequencing. And what this did was it did a lot of things, but the main impact was just it allowed us to collect massive amounts of DNA sequence information with very limited effort. So frequently what we have is very short reads that we are sequencing. These are DNA molecules that are anywhere from nowadays 50 to up to maybe a kilobase or 1,000 base pairs in length. And the way that in brief, we'll talk about that more in detail, but essentially we chop the genome of interest up into small pieces. We attach adapters to the ends of these small pieces. Those adapters then go on what effectively is what we call a flow cell, which is more or less like a glass slide where they bond, and then we do fluorescence chemistry and imaging on all of these little tiny clusters of DNA that grow during the course of the sequencing run. Now, we've now advanced beyond some of the super short read sequencing in certain applications, although as I'll tell you, the Illumina sequencers that I'm most familiar with, still we're using 50 to 100 base pair reads, and we're getting a lot of information from that. However, there are what we call single molecule real-time sequencing protocols now. In particular, the PacBio is leading this, which allows you to get much longer reads, even more than a kilobase in length. So I told you there's many. It's high throughput. It's low cost on a per base perspective. However, the cost is now backloaded onto the bioinformaticians that have to analyze all these massive amounts of data. So I thought I'd start with probably the workhorse machine that is pumping out the vast majority of the human genomes that are being sequenced, and including the massive amounts of RNA-seq data, in which scientists are now using to measure gene expression at the individual cell level and under many, many different conditions. I thought I'd give you a sense of what the HiSeq is, and a little bit about some of the numbers to give you a sense of when I say high throughput, exactly what do I mean. So we have two flow cells. Once again, these are basically like a small glass slide about the size that you would put under a microscope to look at cell preparation. And on each of these is what we call 8 lanes. In a typical run of the HiSeq, on our own machine at NYU, we're getting about 350 million reads per lane. You know, at 100 base pairs per read, you can do the math. That ends up being about 35 gigabases per lane, or 35 billion base pairs. Which, if you do the math, then on a flow cell, you can get around 280 on a good run, gigabases of sequence per flow cell. Remember, there's 2 flow cells per machine, so you can double that if you count on a machine basis how much throughput you can get. Now, for perspective, the human genome is 3 gigabases. That is the haploid complement, or the complement that you would inherit from one parent. It's about 3 gigabases in length. So extrapolating from that 280 gigabases, dividing that by 100, you're getting almost 100-fold coverage. So divided by the genome size, 280 divided by 3, we're getting close to 100x coverage from one flow cell of a high-seq run. And that's what we mean by high throughput. I'll talk in more detail exactly what I mean by coverage in a second, but just to introduce you to some of the other machines, you certainly don't need to memorize this kind of thing, this kind of information. What I'm looking to do is to always give you some perspective on what's out there. Some of these other machines have uses exactly like the HiSeq. So the HiSeq's competitors is mostly solid, and perhaps the PGM machine, which are shorter read technologies that are pumping out similar amounts of data. The solid, in particular, I have some experience with, is known for its accuracy. 454 there. This is the technology that was used to first sequence Jim Watson, one of the co-discoverers of the DNA molecule itself, or the structure of the DNA molecule itself. He lives out on Long Island, and 454 approached him about sequencing his genome. Of course, he was on board with that. And so 454 is a slightly longer read technology. I think it's around 400 base pairs is a typical read length. And so this is paired with HiSeq data, which is a lot of very short reads, say 50 to 100 base pairs. You overlay that with 454 data, which are longer reads, and this allows you to do better things like de novo assembly of genomes, like we'll talk about. So sometimes people combine runs from different platforms to try to maximize sort of be most effective at solving the problem that you're trying to solve. It's not that one of these machines is necessarily better than another. It's really which of these machines are best for your purpose. Just briefly, there are these benchtop systems now. These are becoming almost like things that you have around the lab, like you might have a thermal cycler if you're familiar with PCR. Common, it's not, I would say, common at this point, but this is where the field is heading. This allows people to do on a laboratory technician in an afternoon to set up libraries in the morning, run the sequencer overnight, and the next day you have your set of exome sequences, for example. We'll talk about exomes in our next lecture. Anyway, so this is benchtop systems are much cheaper, much faster. They give you a lot less data, but they allow you to do it quickly. I forgot to mention that a typical HiSeq run can take quite a while. So here, if we actually look here, so let's look at the runtime. We'll start there. Notice that a HiSeq is 11 days. Okay, so that is running a paired end run on one flow cell take about 11 days, whereas some of these benchtop machines can proceed much faster. Let's look at the cost of the machine itself. These are not cheap. You can't buy one for your home or one for your friends. It's $654K was the price. Went for the 2000. We're up to 2500 now. Here's the amount of sequence, 600 gigabases. That's what they quote on their website. I quoted you about 280 for a flow cell, so twice that is 560. They're saying 600 for both flow cells. We're getting close to that in practice. Here's the summary of the read lengths. Here's the MySeq, which is the benchtop one. the Illumina's high-seq here, also maximum 150. Notice I didn't have the 454 here, so these are shorter read lengths than the 454 can provide. Paired data, PacBio does not allow you to do paired end data. Well, if you don't know what that is, I'll tell you in a minute. And then here's the other thing I wanted to mention was the sequencing cost per gigabase. That's $41. So, if the human genome, 3 gigabases gets you 1x coverage. So that's like $150 is 1x coverage. So for 100x coverage, we can, multiply 150 by 100, and somewhere around the ballpark of $10,000 gets you a reasonably deeply sequenced genome, which is truly, a remarkable feat that we've, as a industry, sort of accomplished over the last decade or so. So I'd like to jump now from the machines, the workhorses in our field, to talk a little bit about what do I mean by a reference genome, and some of the common terminology that we'll talk about throughout the course. So a reference genome is really a representative genome of a species. It's not typically generated by next generation sequence data, at least the best reference genomes aren't, because just so you understand the history, if you don't know, for a long time we did what we call Sanger sequencing. Sanger was the guy who discovered the original fluorescence, not the fluorescence, but the biochemistry of the terminators, the terminators that allow you to do the sequencing that we used to do. And this clone-based back sequencing, back means a bacterial artificial chromosome. These are really long inserts in a clone library where you just sequence the ends, and you do what we call shotgun sequencing. You have the ends of a very long molecule that might be 100 kilobases long in length. But the key thing about that back-end sequencing is that you have the sequence of these two fragments at the end of this very long chunk of DNA, and so you know that they're linked. Okay, this is the problem in getting nice, long chromosomes of sequence, which you don't get when you just sequence from a short read library, as we'll discuss. So the genomes that we call of reference have been sequenced with this careful technology. The chromosomes are typically, assembled or near assembled, and they've been done carefully and slowly, and some of this can take 10, 15 or more years of an entire research community in order to build, a really high quality reference genome. The genome is typically we refer to as the haploid complement, as I already mentioned. So we're just taking sort of effectively, what you would inherit from one parent. I should mention that it can be a consensus of many individuals. It's not frequently. It's not just a single individual that is being represented in that reference genome. It's high quality sequence. It can be closed as some of the better sequenced genomes, like in plants and Arabidopsis and rice, and in humans, the genome is virtually closed. Or it may consist of many contigs or scaffolds. So, like, just for an example, I'm working on a project on the date palm, which is a plant, a major crop in the Middle East, where I work with my colleagues at NYU Abu Dhabi, and we're sequencing 100 date palm varieties, what we call the 100 Dates Project. But in this project, we're using a reference genome that consists of 57,000 pieces, okay, little bits of sequence that have been assembled. So from a 600 megabase genome, 57,000 contigs or small bits that have been assembled by next generation sequence data. So this genome was not produced by the careful Sanger-based sequencing. It was produced by sequencing short reads and assembling them. So you get bits like this when you don't have the careful Sanger sequence strategy. So what is a reference used for? Well, I'll describe that here in a second. It's a backbone that facilitates the assembly of closely related genomes. And I'll talk in more detail about that in a second. So let's look at the human reference genome. This is the current release GRCH37, put together by what's called the Genome Resource Consortium. It's actually derived from 13 people from Buffalo, New York, whose DNA was sequenced and then combined to form a consensus. There's 22 autosomes in the human genome. There's the X and Y sex chromosomes, and last but not least, there's the small piece of the mitochondrial genome, which is only 15 or 16 kb. That's thousands of base pairs, so it's a very small piece. But these are the molecules that make up the human reference genome. It used to be quite broken up, where there was 150,000 gaps. These are pieces where we don't know how everything fits together. But as of 2010, we're down to 250 gaps. So it's basically closed. It contains some unlocalized and unplaced or alternate loci. Alternate loci are basically copy number variants, pieces of DNA that might exist in one individual but might not exist in other individuals. And I just thought I'd alert you to the fact, because many of you someday will be working with the human reference genome, that there are different sort of versions of it. So different people have tailored it for their very specific needs after the consortium has released it. So you just have to kind of pay attention to exactly what version of the reference you're downloading when you're doing your analysis. And good news is I've just found out that the build 38, this will be GRCH38, should be out later this summer. So what do we do with the reference genome? Well, as you probably know, the sort of standard protocol is to sequence your short reads, either RNA-seq or DNA sequencing, and then map those those reads back to the reference genome. So the reference really becomes then a guide or a backbone on which you can map closely related sequence. It has to be closely related, so it wouldn't work too well to map human DNA to chimps, although we're not that diverged from chimpanzees, only about 1 to 2%. It would work just fine, but the closer related genomes, like other human individuals, would work much better, because on average humans only differ from each other at about one position out of 1,000, not 1 out of 100 compared with chimpanzees. So we use it for RNA-seq studies of gene expression, where we map, so we sequence mRNAs. Each read then can be mapped back to the reference. We use it for whole genome resequencing. We'll talk about this quite a lot in this class. Re means that we're sequencing the genome of the same species again and again and again, just from different individuals. Okay, so this is the field of population genomics. Resequencing, I guess, could also be encompass cancer applications and clinical applications where people are sequencing tumors and comparing them back with somatic tissues, although maybe we can refer to that as something else. But today we'll sort of lump it into this whole genome resequencing, and then of course we can use the reference genome to assist in assembly of a new genome of a divergent species. So right now I'm working on another project where we're assembling, with some of your classmates actually, where we've sequenced a close relative of an algal species that is we're interested in for biofuel purposes, and what we're doing is we're trying to use a studied reference genome. It's about 20% divergent, but we can still get some of the reads to map, and we're hoping to use that to assist in our assembly, to at least get some linkage relationships between contigs that we otherwise had no idea where they are. So let's briefly talk about paired end versus single end libraries. So when we do the sequencing itself, we build libraries. Okay, so the libraries themselves are really consist of basically adapters that have been put onto a the ends of a fragment of genomic DNA, and in some chemistries circularized, and in others not. But then what these libraries are then put on the flow cell after doing a number of biochemical steps. But whether we paired end versus single end is essentially whether you have adapters on both ends or an adapter on only one end of the genomic fragment or RNA-seq fragment. And in the end, for a paired end read, we sequence in off of both adapters at either end, but for single end, of course, we only are sequencing off of one of the adapters. So single end, you don't get the paired information. So in this diagram, maybe it'll help. Here, the blue part is not sequenced for each of these clones, where the red parts, the adapters, are sequenced. So here, this clone would be sequenced twice on this paired end read, but the blue part in between the red would not be sequenced. So red gets sequenced, blue does not. So why do we do paired end, or why do we only do single end? Single end's cheaper. You only get half the sequence, but sometimes you don't need the pairs. So in RNA-seq applications, really you just want to tag the RNA molecule that you're sequencing one time. So you want to sequence each RNA one time, and you don't need the second pair in principle to just quantify. But in many other applications, we're not just interested in quantifying how many mRNAs that were there. So in another gene expression sort of application, we want to infer the alternative splice variants. So these are where exons are from the same gene. There's a different mix of exons used for different mature mRNAs, and sometimes you don't know which of these alternate splice variants is being used. So many human genes are alternatively spliced. So you can figure out if you know that, say, this end of the read is in exon 1 and this end of the read is in exon 2, then you know that this particular transcript must have consisted of exon 1 and 2 and not, say, exon 1 and 3. Okay, so this allows you to do this alternative splice site detection, or splice variant, detection. It allows you to detect structural variants. I won't go into detail now. Of course, de novo assembly, if you remember my example from the back ends, it's very similar, right? In the back ends, we had like 100 kb of sequence in between our two ends that we've sequenced and no sequence in between. Here we have very short reads, or clones. So these, in Illumina technology, these are like 300 to 400 bases apart. So the blue part here in this diagram is like 300 to 400 bases. We're sequencing 50 or 100 on either end. So we do get some linkage relationships. We know these molecules are sitting on the same, are from the same chromosome. It's just that they're very short. So when we do de novo assembly, we can't get get the really long contigs that we'd like to. So there's a few other details here why you do paired end. Maybe we can talk about this later as we move along in the course. But briefly. It improves mapping rates. So if you know that the one on the left here maps to one position in the genome, then and maybe this one doesn't map as well, say because there's a mutation that differs from the reference, so it doesn't map as well. But because it's right next to it, then both of them will map. So that's actually what we call mate rescue. Improved mapping rates I guess that's really the same thing. Mate rescue means that if this one maps really well, this one doesn't map so well, because this one maps so well and this one sort of mapped, then the aligner will say that both of these mapped. And so that's mate rescue. So this is a mate. So this is one read and this is its mate read. And in the end, it also leads to increased mapping quality scores. So the quality scores are aware of whether or not the mates mapped or not. So we'll talk about what map quality scores mean later. So one final thing here. Here I'm telling you about how much sequence we get when we do. This is the terminology 2 by 100. So this means that a 2 by 100 is when you have paired end. So 2 means that there's two red ends that we're sequencing. 100 means the length of each red end by itself. So any one fragment, the whole thing here we call a fragment. Okay, some people will call it the whole thing a read. I just want you to be aware of that. In this class, we'll call each red piece a read. Okay, the whole thing we'll call a fragment. So both ends, or both paired end reads, we'll call a fragment. So 2 by 100, that's 200 base pairs per clone, or per fragment. Yeah, here I'm just giving you some numbers about, how much, how much. I said 400. I think the last slide I said 350 for about how much we can get in a single lane. So this is 1 by 100 is a single end type approach. Okay, so let's move along. Library indexing or barcoding or multiplexing, all of these terms refer to the same concept. This is where when you have a lane, but you don't just have to sequence one library per lane, you can sequence many libraries per lane. So the way that you sort out which sample is which when you've sequenced many libraries per lane is you add what we call a barcode to the adapters, okay, that are sitting at the ends of these fragments, okay. So the adapters get sequenced, so we prepare each sample separately as its own library, and each sample gets a separate barcode, which then becomes the sequence that allows us to distinguish that sample after the fact. So we throw all of the libraries together on the same lane. They all bind to the same flow cell. The clusters that form on the flow cell, as we'll talk about in a minute, all form, intermingled. But what happens is that bioinformatically at the end, we use computers to tell us which cluster or which sequence came from which original sample. Okay, so that's the concept of barcoding. So we can sequence in many applications. We're not interested in massive coverage because we don't need it to address the scientific question. And so what we do is we index or we multiplex different samples onto the same lane, sequence all of the samples at lower coverage. So essentially you can take the numbers that I quoted to you for the numbers of reads, say 350 million per lane. If we sequence 10 samples in 10 different libraries, that would be 350 million now spread across 10, so we'd expect about 35 million per sample or per library. So again, we use the barcode for computational demultiplexing after the sequencing run is done. So this part is not terribly important for the purposes of this class, but it always helps to know a little bit about what's going on. So for example, when the first thing that's really important to know about the cell is that when it comes to RNA-seq applications is that the cell is loaded with this stuff called ribosomal RNA. These are the RNAs that go into the ribosome for translation or the production of protein from an mRNA template. And more than 50 to 90 or even more perhaps percent of all of the RNAs in the cell are ribosomal RNA, and they're not interesting from the perspective of a biologist. Biologists are interested in the 20 or 25,000 what we call protein coding genes, not the ribosomal RNA coding genes. And so we need to get rid of all of this stuff, which is making up the bulk of the RNA, because we don't want to sequence this stuff because it's not interesting. So how do we do that? We use what's called polyase selection. Don't worry about it. Just the concept's important. So if somebody gave you some RNA-seq data, you might ask them, how did you? First thing you might ask for is, how did you get rid of the ribosomal RNA? How did you deplete it of ribosomal RNA? So what we do then is we share it up into little bits. Little pieces. We size select. Again, remember I was telling you about 300 to 400 base pairs is the insert size for Illumina libraries. We ligate on the adapters onto the end the same way we do DNA. I think I actually skipped a step here. We actually do a step here where we convert the RNA to what we call cDNA, so then we can, which is not terribly important for our purposes. We can then, but we're going to end up sequencing a molecule of DNA. It's just that because DNA and RNA are complementary, we can infer what the RNA was when we sequence the DNA. We PCR amplify to get more of it, which is an issue in RNA-seq analysis, because really what we want is to sequence one molecule on our flow cell, which represents one molecule of RNA. When we PCR amplify, now we're amplifying many molecules. We're creating many molecules from one original RNA molecule, which is, then can be sequenced many times. So just be aware that this, can cause, if some molecules amplify better than other molecules, then that can lead it to some bias in the quantification of the original relative proportions of the different mRNAs which we're trying to infer. Quantifying libraries using qPCR, just know that you need to put the right amount of DNA onto a flow cell in order to sequence it, so we have to quantify the libraries carefully. So these same techniques are, here, just the ribosomal depletion. There's also the step, which I forgot to put on here, with the cDNA production, which are unique to RNA-seq. DNA sequencing follows a very similar protocol. So how, in the next few minutes, let's just maybe close with how does the sequencer itself work. So I'll leave this to you to go, to understand to whatever detail you'd like to know. But for my purposes here, what to some extent, you just understand that the Illumina isn't terribly, willing to give you the entire detail here, because this is their proprietary chemistry and how it works. So, we can understand it sort of at a basic level, but we don't. Even I don't understand it in extreme detail, simply because it's actually, when you start digging, it's hard to get the exact details of the chemistry. But essentially, remember, let's imagine a DNA experiment. We have the chopped up DNA fragments of, say, 300 to 400 base pairs long. We then ligate these adapters to either end. We know the adapter is a forward and a reverse, okay? And those are indicated by the pink and the purple here. We quantify all of these each library. So in this experiment, let's imagine that this is a single sample from a single cancer cell, and we're going to put one cancer cell DNA preparation on a single lane. Okay, so or actually, yeah, let's call this a lane for our purposes. And so what happens is these adapters actually ligate to a molecule that's already stuck to the glass flow cell. Okay, so this molecule, a complement to the adapters here, so you denature the DNA, and what happens is that they're not showing all of the detail here, but you get complementary base pairing, okay, between these fragments that are stamped already onto the flow cell, and they bind to now the library pieces. Okay, so now a bound one now is sticking straight up like this, and so what happens is the sequencing then starts off of the primer that's bound here. Okay, so the bound piece is complementary, and it has a free 3' end, which can then be extended. Okay, and what happens is that fluorescent dyes, okay, during each cycle, okay, are extended, and what happens then is that an image comes in, and the images, or takes a picture of all of the different clusters. Okay, there's a cluster here, there's a cluster here, there's one over here, and the imager, it looks down on this flow cell, takes a photo of the fluorescence, and it is that provides the signal at one position in the read. So each one of these clusters, okay, after the run, will provide a paired end sequence. Okay, So the paired ends, this is one end and this is the other, and we start with the forward read. Okay, so again, for one cycle of the run, the A's, T's, G's, and C's, which have maybe on one cycle, the A's have greens, the T's have reds, fluorescent dye. So they complementary base pair right at the end of the exposed 3' end of the primer. The imager comes in, takes a sequence of this, a picture, excuse me, and each of these clusters then has a color associated with it for that cycle, indicating what base is in that particular cluster at that position. So the first cycle represents the first position after the primer. Second cycle represents the next position, and again, so you wash off all the dye essentially, and the nucleotides associated with those dyes, and then you go to the next cycle, and again allow things to hybridize and take a picture. Okay, so that second image then gives you a different set of colors representing the second position. Okay, in the newly sequenced read. Okay, so what they're showing here is how do you sequence the second read, and this is really the tricky part, because you don't have this nice primer attached here that provides this 3' exposed end in which you can then bind new second strand nucleotides to the template DNA, which is the thing that we're sequencing. And so what happens here is that we have what's called bridge PCR, and this is where it gets really dicey about how it works. But after you finish sequencing the first read, the molecule of DNA, from our original library, can flip over and it can bind. If you notice, it was purple here. Now it goes over and binds to the purple primer that's stamped onto the glass slide. Okay, then it somehow cuts itself free, and you end up with having flipped molecules here. So this is where it's like crazy. Like when I first read this several years ago about this, it's like, wow, that's pretty slick chemistry. That's the part where things really break down for me. But effectively, what you do, you flip the molecule over. Now you have a free 3' end here, and you can go through the same process, in the second stage where you're sequencing the second read. So again, each cluster gets sequenced base by base, where fluorescent nucleotides bind. An imager comes in, takes a picture of what are millions. So each cluster, remember when I tell you that there's 350 million reads, that's single end reads, so that would be 175 million fragments, or 175 million clusters. Each one of these is a cluster that will yield the sequence of 2 reads, a forward read and a reverse read. I hope that's clear. You'll have a chance to ask me any questions you want to ask me. I want to give you this. It's important to understand this, because there's some technical issues that you have to deal with bioinformaticians to kind of clean up some of this data. Hopefully, when it comes off of this flow cell, because there's imaging problems, clusters get merged. They kind of grow like amoeba as the sequencing goes on, and they merge together. So bioinformatically, we have to pull those clusters apart, and there's good ways to do that. But anyway, it helps to know a little bit about the technology. So why don't we call it quits there? Everybody take a break, and the next, you can look at there on the next lecture, which will be sort of, a second, stage introduction to next generation sequencing. So check that out, and I'll see you next time.